Good morning, good afternoon, and for some of you, good evening. Warm welcome to all of you to this Bioaster IABS webinar. I am Joris van der Putte, the president of the International Alliance for Biological Standardization, and I have the big pleasure to welcome all of you. The International Alliance for Biological Standardization, IABS, is used to convene, to bring people together, to openly discuss and come to consensus in order to progress with innovations. And we are so happy that this innovative approach is part of our common webinar, Bioaster, who is also a very innovative organization. And us, IABS, more than 65 years experienced in the bringing together and taking up challenges for the future. I like to address in my introduction a very special warm word of welcome and congratulations to Professor Jennifer Vargo for her World Living Legend faculty achievement, a big award in basic research at the University of Texas in the Cancer Center. So Professor Vargo has a long, excellent career in understanding treatment of diseases through science. She's a specialist of immunotherapy in cancer, particularly melanoma, and she also did a lot of groundwork, high-level work on adjuvant, neoadjuvants in the use and blockade of cancer. And she's a globally, worldwide recognized leader in the impact of the gut microbiome on cancer. So, Professor Vargo, we are so happy you are part of our discussion and that you are actively presenting and giving your views in this quite new avenue for health. So Professor Vargo has numerous awards. I will not talk about all of them, but you all will believe me that she is a very highly recognized and proves her multiple hundreds of papers published in Nature, Cell, Science, etc., etc. So we as IABS, of course, we are always very happy to have the best scientists of the world. And I will now introduce the first speaker, Professor Cyril Guillard, who has also global recognition. He got his PhD at less than 70 kilometers from my home here in Lille. And he has a master at the University of Strasbourg before taking up a high-level scientific career at the NIH in the United States, before then moving, if I'm correct, to Toronto at Sinai Hospital and in senior research positions in Toronto, before joining the very fantastic organization Bioaster, who is really a kind of jewel of biotechnology, not only in France, but over whole Europe. So, Professor Guillard, I leave the floor to you, even 1 minute before you had to start.
Thank you.
All of you.
Thank you, Joris. I think you're too nice. Actually, I was in Toronto. I was a scientist at Mount Sinai, but my principal employers was Public Health Ontario at that time, where I was a primary investigator. That's the Mount Sinai Hospital was a cross appointment, but let's not talk about me. That's not the point today. I think I'm glad to see the large numbers of attendees. It's growing up, and we have a lot of people that signed up for this event. I think it's pretty cool because it's a new topic. It's a new topic for regulation. It's outside of what I believe from what IABS uh, uh, expand or discuss usually in his meeting, and I think it's good to see those brand new topics coming up. And fecal microbiota transplantation is one of them. Today we've gathered uh, uh, speakers and a group of speakers covering this topic from the clinical, biotech, and regulatory point of view. It's right in the heart of the mission of IABS. It's also in a mission of Bioaster, not the regulation part, but the technologies related and the bridges between the biotechs, the innovations, the basic research, and in the presence of those speakers today is a good demonstration of this. Just few words on Bioaster because you'll get more information later on with the presentations of Vincent Thomas. At Bioaster, we have 4 research programs, and we are a non-for-profit technology research institute. And among those 4 programs, we have our microbiome programs. This one is very interesting to work for in the technology field because it's super transversal. It goes to usage in the diagnostic field, also in the treatment field, infectious disease, and also in the cancer field. That's why it's a super exciting field for us, and it brings us a lot of collaborations with brilliant companies, and among them the one that that will be presenting today. We've been working with Mad Pharma in the past, and I think I hope Hervé will say it was a success. It was a success. So it's really nice to be with you. I have 3 minutes left. I don't think I'm necessarily going to use those 3 minutes to talk about Bioaster or me, but I'm going to use them to introduce Dr. Sergaki, who's our next speaker. I hope I said it well, Dr. Segaki. I'm going to say Chris next after that, if it's okay for you. So Chris is a scientist in the microbiome sections at the National Institute for Biological Standards and Control. Chris is working on microbial interactions and how to use them to the benefit of human health. In our institute right now, she's leading a project on the role of the microbiome in gut health and disease. In addition, she's leading a development of reference reagents that can act as a global standard with, and those standards will be evaluated as candidate for the first World Health Organization wholesale reference reagent for the microbiome. So it's just in the perfect topic for our discussions today. And this project aims to set light to the bias introduced by the DNA extractions process in the microbiome research. Chris is also leading, well, you have a really long sketch. He's also leading a global World Health Organization collaborative study for the establishment of the first World Health Organization DNA reference reagent for NGS. And before joining our current institute in 2019, Chris completed her PhD at the University of Warwick in UK, researching microbial interactions in fungi and bacteria, and their impact on the host by combining a laboratory and bioinformatic approach. I think we're almost there, Chris. It's almost 10 after 3, so you can start in a few seconds. The floor is yours.
Thank you very much, Cyril. Very kind of you. Let me share my presentation. I hope you can all see it.
Yes.
Yes.
Perfect. So thank you very much. I'm really excited for being with you today. My name is Chrissy Sergaki. I know it's not the easiest name of the world. I work at IABS in the UK, and I will present our work on the developing global standards for the microbiome field. So for those who don't know us, IABS is part of the Medicines and Healthcare Products Regulatory Agency in the United Kingdom. IABS is the UK's official medicines control laboratory. It is also a World Health Organization international laboratory for biological standardization, producing more than 95% of the WHO standards. And we also perform applied research to ensure the safety of biological medicines. So the last 10 years, there's intense research around the human-associated microbiome and its role in health and disease. Particularly work on the gut microbiome has shown that the gut microbiome has a key role in a lot of different key functions of the human body, such as protection against pathogens, synthesis of vitamins, immune system development, promotion of intestinal angiogenesis, promotion of fat storage, short-chain fatty acid production, and modulation of the central nervous system. And exactly because gut microbiome has such a key role in so many important functions of the human body, when there is gut dysbiosis, so when there is a disturbance or a change in the density or the composition of the microorganisms within the gut, it has been associated with a lot of different disease and conditions of the human body, such as musculoskeletal, pulmonary, digestive, of course, cancer, cardiometabolic, and diseases and conditions of the nervous system. And because it plays such an important role in so many different diseases, there have been a lot of efforts to develop therapeutics involving the gut microbiome. One of those is the fecal microbiome transplant, or the FMT, which is the only microbiome therapy available, and it uses fecal material from healthy donors to treat patients. Clostridium difficile infection is the first and only condition to be widely treated by FMT, and it has shown remarkable efficacy of 92%. But besides these, there are 146 active clinical trials using FMT for a lot of different diseases like antimicrobial resistance, conditions, cancer, hypertension, diabetes, irritable bowel disease and irritable bowel syndrome, cirrhosis, multiple sclerosis, allergies, asthma, epilepsy. 46 of those clinical trials are in phase 2, and 18 of those are in phase 3 clinical trials. There are also the live biotherapeutics products, or LBPs, the so-called designer probiotics, which may replace FMT in the future, although the development is slow. One example of these LBPs is the EcoSport 3 from Sears that just finished the phase 3 trials with 30% reduction in reoccurrence of patients with C. difficile, which is already reduced efficacy compared to that of FMT. And while there is so much research around the gut microbiome and gut microbiome therapeutics, we know that there are no WHO endorsed reference reagents available for the microbiome field. There are a lot of studies that highlight the variability across methodologies, and we know that in the microbiome field, we have really big problem with reproducibility and comparability between studies. Because of this, IABS launched the microbiome program in late 2016 to investigate the need and requirement of WHO reference reagents for emerging microbiome therapeutics. Since then, we're leading 18 WHO endorsed projects to establish WHO reference reagents for the microbiome research. So the big challenge when developing reference reagents for the microbiome field is that there are really a lot of steps from sampling a microbiome from a body side until we have the profile of the microbiome in that sample. as you can see here in the slide. In order to overcome this challenge, we broke this process down to 3 parts, which we think introduce different type of bias. And these 3 parts are collection and storage of the sample, DNA extraction of the sample, and sequencing and bioinformatics. We started the development of the reagents from the bottom, from the sequencing and bioinformatics. When started this work, we knew that the microbiome comprises more than 1,000 species. Many of them are unknown and they can vary massively across individuals. So we had 3 big questions. Which strains should we include in the reagent? How should we measure them? And how will these reagents be used in the real world? Our approach to this was by developing DNA mock communities for key different microbiome sites using reference strains. So strains most commonly found in each body site. We then developed the 4 key reporting measures for data generated by NGS assays. And these are sensitivity, which is the percentage of correctly identified strains, the false positive relative abundance, or FPRA, which is the relative abundance of the false positives, the diversity, which is the number of observed species, and similarity, which is a breaker to similarity between known composition and observed composition. So similarity essentially includes what do we have in there, with what species, how many, and what relative abundance. And the way we think that this can work in the real world is by setting an acceptable range of its measurement and set go and no-go criteria for the users. So, as I said, we developed DNA reference reagents for different body sites. But for today, we will focus to the gut DNA reference reagent because it's the most relevant to the FMT. As you can see, here is the list with the 20 strains we included in the reagent. They comprise of 5 phyla, 13 families, 16 genera, and 19 species to really challenge the ability of the pipelines at different levels. What we did is extracted DNA from each strain individually. We QC this, we made sure that everything is pure. And we combined them together, creating mix and high-low variants. Mix means that all strains are found in equal ratios, and high-low variance means that the strains are found in staggered ratios. Again, to really challenge the ability of the pipelines to correctly identify strains of very low abundance. We then tested these DNA reference reagents with shotgun and 16S RNA sequencing and analyzed the sequencing data with different bioinformatics pipelines. Here we'll present only results from the shotgun sequencing, but you can find more information in the publication. So on the left-hand side, we have the results from the gut mix reference reagent, on the right-hand side from the gut high-low. I don't know if you can see my mouse. So if we look at the left-hand side, the first bar represents the actual microbiome composition, and the rest of the bars represent the microbiome composition in the reagent as identified depending on the bioinformatic tool we use to analyze the sequencing data with, for example, Metaflan2, Kaiju, Kraken, Bracken, and Centrifuge. And you can see in both reagents, we have really big differences in the microbiome. And to really be able to compare these differences, we use the 4 key reporting measures: sensitivity, FPRA, diversity, and similarity. And you can see really well here that we have really big differences in all 4 measures. So if we have a closer look, for example, Kaiju had 100% sensitivity, which means that it was able to identify all strains present in the reagent. However, diversity was 158 instead of 20. So we expect to have 20 strains in the reagent, but Kaiju identified 158. And you can see from the FPRA is 10%, which means we have 10% false positive relative abundance. And that practically means when we have a clinical sample and we try to see whether there is gut dysbiosis or not in there, if there is gut dysbiosis and we actually have 20 strains in there, but because of the pipeline we use, we massively overestimate the number of strains present in the sample, we may reach inconclusive results. On the other hand, if we have a look at the metaflan 2, the sensitivity was lower, was 89%. However, the diversity was 70. So it was able to identify 17 out of the 20 strains, but this was all accurate. So you can see from the false positive relative abundance is 0. So even though it wasn't able to identify all the strains present, the ones it identified was correct. And with that, we saw that the reference reagents are capable of evaluating pipelines with a 20 strain mix. We then wanted to test whether the pipeline performance is influenced by the reagent composition. So we used the NMBSE reference reagents along with commercial reagents from ATCC and Zymo. We sequenced them together and we analyzed them together with different pipelines. And what we found was that changing the reagent changed the performance of the tools. However, we found that reagents of low complexity had significantly higher measures of sensitivity and similarity. That means that when you use a reagent of low complexity to study an area that has high complexity microbiome, you may find that your pipelines perform perfect, just because the low complexity reagent is not able to really challenge the ability of the pipelines to correctly identify everything that is in your sample. So it's really important to use a reagent that reflects your intended area of study. We also tested the stability and homogeneity of the reagent. We performed an accelerated degradation study. We placed our reagent in different temperatures from -70 to +56 degrees Celsius. And we studied over time. And we found that the reagent is stable over time with 98% similarity when comparing between time point 0 and 6 months at room temperature. And that's the same 12 months after that we have the data now for it. So the reagent is quite stable over time. Once we developed the DNA reference reagents to standardize the sequencing and bioinformatics step of the process, we moved to the DNA extraction part of the process. For this, to standardize the step, we developed the wholesale reference reagent that contains the same 20 strains as the DNA mix reference reagent. So again, it comprises of 5 phyla, 13 families, 16 genera, and 19 species. They're all present in equal relative abundance. The reagent contains 2 times 10 to the 9th cells per ml, and the material is lyophilized. The big challenge when developing the wholesale reference reagent where the stability, we had to ensure that the reagent remains stable over time. So the cells won't start lysing, they will start killing each other or multiplying. We of course had to make sure that we can maintain the integrity of the cells, because we need the whole cell form in order to really challenge the ability of the DNA extraction kit to break it. And we needed to ensure the accuracy of the cell enumeration. So we needed to make sure that we can accurately count the cells in the reagent. Our approach to overcome these challenges was by fixing the cells. And of course, when fixing the cells, the most important challenge is to make sure that we maintain the whole cell form with the DNA inside them. And we validated this using confocal microscopy and cryo-electron microscopy. As you can see here on the left-hand side, we're able, with confocal microscopy, we're able to see the cells with the DNA inside them. We're also able to see RNA inside them, which could be interesting for potential future applications. You can see even more nicely with the cryo-electron microscopy pictures that the cells they maintain their whole cell form. And I don't know if you can see here, we can observe the DNA swirls. So the DNA is still inside them. And this is quite obvious in the bottom left picture as well, with the DNA swirls in the middle of the cell. So once we made sure that our method works and maintains the whole cell form of the cell with DNA inside them, we try to find the best way to accurately measure, count the number of cells in the reagent. We tried, of course, microscopy, microflow imaging, but we found that flow cytometry gave the most accurate and reproducible results. As you can see here, the coefficient of variation was quite low, so we had really reproducible results with flow cytometry, so we went ahead with that method. We then used the whole cell reference reagent to extract DNA from it through 8 different commercial DNA extraction kits, and most commonly used in the gut microbiome research, from 5 different companies. And as you can see from the table here, we tried to have DNA extraction kits with different approaches to the DNA extraction process to really see how that affects the results. With that, we were able to assess the extraction efficiency and quality of the DNA extraction kits. And if we look at the second column, you can see that we had really variable yield from 125 nanograms of DNA to 2,400 nanograms of DNA. We also measured the integrity and purity, but they were quite comparable between the kits. So with that, we saw that the whole cell reference reagents allow for assessment of the DNA extraction efficiency. We, of course, use the DNA to sequence it with both shotgun and 16S rRNA sequencing. However, here I will only present the shotgun sequencing results analyzed with metaflan-3. Again, the first bar represents the actual microbiome composition. The second bar represents the microbiome composition in the DNA reference reagent. And the rest of the bars represents the microbiome composition as extracted from the different DNA extraction kits. And again, you can see really big differences depending on the DNA extraction kit we used. So to have a better look on the numbers, we can use the 4 reporting measures: sensitivity, FBRA, diversity, similarity to the actual composition, and to the composition of the DNA reference reagent. If we focus on the similarity to the actual composition, you can see here that there is a 25% reduction in similarity to the actual composition in the DNA reference reagent. So 25% reduction due to the bioinformatic bias. And there is a further 10 to 20% reduction in similarity to the actual composition due to the DNA extraction process. So altogether, there is a 35 to 45% reduction in similarity to the actual composition. So it could be 35 to 45 reduced accuracy when we're looking in real actual microbiome samples. We also tried different combinations of pipelines and kits, but this did not increase the performance of one another. And with that, we saw that by combining reagents, we can begin to better understand microbiome performance and current limits of accuracy for pipelines used in microbiome analysis. So I presented how we developed the DNA reference reagents to standardize the sequencing and bioinformatic part of the process, the development of the whole cell reference reagent for the standardization of the DNA extraction part of the process. And in the future, we aim to develop matrix reference reagent to standardize the collection and storage part of the process. To conclude, the NIBSC reference reagents can analyze and evaluate microbiome analytical pipeline performance at different steps. And I want to highlight here that we don't want to recommend or to reject any of the DNA extraction kits or the bioinformatics pipelines used in the presentation or in our work. We want people, scientists, to take these reagents and really optimize the methods they currently use. And also, of course, the developers or the manufacturers to use these reagents and support the development of new pipelines, more accurate pipelines that actually can help the microbiome field grow faster. Also, it is important that these NGS assays and DNA extraction kits need to be first validated to be fit for purpose for studying the microbiome using these defined ground truth reference reagents before we can start testing for reproducibility in clinical material. And in order to achieve these, we need to establish consensus thresholds for users to ensure a global harmonization. To do that, we launched a collaborative study to establish the DNA gut reagents as WHO International Reference Reagents for NGS analysis. We have recruited 24 leading labs of the field from 4 continents. So this is a global effort that will test the DNA reagents and assess inter-laboratory variation in sequencing and assign minimum quality criteria that users must achieve when using these reagents. So it will be an agreement from the community. The results will be published in 2022, so please keep an eye for that. And we aim to initiate more collaborative studies in 2022, for example, for the whole cell reference reagents or for the DNA reference reagents for the rest of the body sites. So please get in touch with me if you would like to be part of the studies. This is a copyright slide. And I would like to thank everyone at NIBSC for their work and their help in the development of these reference reagents. And thank you very much for your attention.
Thank you, Dr. Sagaki. So we got only 3 minutes for questions. You want to shoot on your side? Did you receive questions for Dr. Sagaki? Phil, you on?
We have no questions in the Q&A box at this point, but I encourage people to, if you have burning questions for the individual speaker, to put them in now, or we can circle back and also address those questions in the panel discussion. One question just came in. Thank you. So, Chrissy, how do you account for the role of bacteriophage in the mixtures?
Well, the answer is that we don't. We really start from, I think most of the work at the moment in the microbiome field, or like even with FMT, focuses on the bacteria. So we start from the bacteria, we're hoping to move to other parts or like members of the microbiome, but we tackle each challenge individually, really. So this is only for bacteria.
Okay, thank you. We have one other from David Rhodes, and he wants to know if and how you maintained anaerobic conditions during your whole cell references.
Oh, so I prepare my cells in anaerobic conditions, of course, for those that are anaerobic, but then I fix them, so I kill them. So the cells don't need to anaerobic conditions anymore. And that's again, because if we have anaerobic cells that are alive in our mixture, we have the issue of them lysing or potentially lysing. So if we kill them, we make sure that they maintain their whole cell form and they're not going to lyse when they get to the oxygen. So this is how we tackle this issue.
Okay, thank you so much.
Thank you.
Thank you. So I think we're just on time. Our next speaker is the Professor Ari Sokol. So Dr. Sokol is a professor in the gastroenterology department of the Saint-Antoine Hospital. So it's part of the largest network of hospitals in Paris and the largest network in France. He's the co-director of the microbiota, gut and inflammation team at the Sorbonne University and group leader in the Michaelis Institute at INRA. Ari is an internationally recognized expert in the field of IBD and gut microbiota. He published 240 papers on these topics in major journals. The top ones, I'm just citing a couple: Nature Communications, Nature Medicines. His work on the role of the gut microbiota in IBD pathogenesis led to landmark papers describing the IBD-associated dysbiosis and on the role of the pivotal commensal bacteria Faecalibacterium prosnitzi and in gut homeostasis and in IBD. Currently, his work focuses on deciphering the gut microbiota host interactions in health and disease, with a particular focus on IBD in order to better understand their role in pathogenesis and develop innovative treatments. Dr. Sokol is exploring particularly the role of the microbiota in tryptophan metabolisms, for which he is the recipient of an ERC grant. It's the European prestigious European grant from the European Community. And beside basic science, he's also involved in the translational research. In translational research project, he's the current president of a French group of fecal microbiota transplantations and is the head of French Paris network APHP, a network of hospital on fecal microbiota transplantation center. He coordinated a pilot randomized controlled trial evaluating the FMT in Crohn's disease, and is currently coordinating 2 phase 3 nationwide randomized controlled trial evaluating this approach in colitis, ulcerative colitis, and Crohn's disease. And he's also an entrepreneur because Ari Sokol also co-founded the company Excellium Bioscience, and the title of his talk is Current and Emerging Indication of FMT. Can it really be a standardized treatment? Thank you, Dr. Sokol, for being present today and for giving this talk.
Thank you very much for the very kind introduction. I hope you can see my slide now.
Yeah, this is all good.
Perfect. So here are my conflicts of interest. So I will not take a lot of time to remind you what is a gut microbiota. Of course, it's a very complex ecosystem with bacteria mostly. At least this is the part we know the best. And among these bacteria, particularly in the colon, we have mostly anaerobic bacteria belonging to these 2 major phyla, the Bacteroidetes and the Firmicutes. But we also know now that there are many other types of microorganisms in the gut, fungi, phages, but also archaea and protists. As it was just told you by the previous speaker, the gut microbiota is involved in many physiological functions and also in the onset or severity of several diseases. So the concept of FMT is relatively simple. The idea is basically to replace an altered gut microbiota for which we think it plays a role in a disease by an healthy one, with, of course, the hope to have positive therapeutic effects. Currently, everywhere in the world, there is only one indication of fecal microbiota transplantation in clinical practice, and this is Clostridioides difficile infection and actually recurrent C. diff infection. The efficacy of this approach was shown almost 10 years ago now by this very nice study. And as you can see, in this patient with recurrent C. diff infection, FMT is able to cure the patient in 80 to 95% of the case, compared to 20 to 30 patients using the usual vancomycin treatment. FMT is also evaluated in patients with severe or complicated C. diff infection as an alternative to surgery. We do not have any randomized controlled trial results yet, but there are several data suggesting a very high clinical cure rate, around 90%. All the other potential indications currently are still in the research field. Following this first study in 2013, the results of FMT in C. diff infection has been confirmed in many other studies. There is a lot of reassuring safety data. The processes have changed. We are using now frozen stools and other types of delivery routes, such as capsules. Many guidelines have been published everywhere in the world, and there have been the development of stool banks also in many countries, and this is the case, for example, in France, and particularly in the Paris area. There have been several studies beyond C. diff infection. Here you have a list of few indications for which we have, in some cases, a result of randomized control trial. In other cases, we do not, but we have only preliminary data or open label data. For sake of time, I will talk only about two important indications, I think: inflammatory bowel disease, for which we have results of randomized control trial, and also the use of FMT as a combination with immune checkpoint inhibitors in cancer. So there are two types of inflammatory bowel disease: ulcerative colitis and Crohn's disease. They are affecting young subjects, altering their quality of life. They are chronic disease with period of flare and remission. We do not have a curative treatment. The patient can undergo different types of complication, severe flare leading to hospitalization, but also infections or neoplasias that are linked either to the disease itself or favored by the immunosuppressant we have to give them. The gut microbiota is known now for many years as a crucial actor in the pathogenesis of inflammatory bowel disease. There have been 4 published studies evaluating FMT in ulcerative colitis, and all of these studies have been performed in active patients with the aim to induce remission. I will not go into the detail of all these studies, but globally, 3 of these 4 trials were positive, and actually even the last one also showed a non-statistically significant trend in favor of FMT. This study had very high heterogeneity in terms of procedures, storage of the stools, intensity of the FMT, and so on. But still, globally, there was a positive effect of FMT with 20 to 30% of the patients who achieved remission at 7 to 12 weeks, compared to 5 to 10% in the placebo arm. It can seem a little bit low compared particularly to the result we have with FMT in C. diff infection, but we have to remember that IBD is not a pure microbiota-based disease. And also, actually, this effect size is quite similar to the result we have in early phase with conventional molecules. In terms of limitation, this study had globally a small number of subjects, and we didn't have maintenance data. In Crohn's disease, we published the only randomized controlled study to date with an original concept and design, which is to target both the immune system and the microbiota. So here, what we did, we enrolled patients with active Crohn's disease. We first treated them with corticosteroids to induce clinical remission. And when clinical remission was achieved, we randomized them to receive one single fecal microbiota transplantation through colonoscopy, then or sham transplantation, of course. And then we tapered the corticosteroids and monitor the patient. I want just to tell you that this is a pilot study with a very small number of patients, so 9 patients per group. And so this study is more to generate hypotheses than to really decide whether FMT is active or not in this type of patients. So here are the results. Here, this is a survival, flare-free survival curve. So by definition, all the patients were in clinical remission before receiving FMT because it was mandatory to be randomized. And you can see that in the FMT arm, the relapse rate is lower than in the sham transplantation group. Of course, there is no statistically significant differences because the numbers are very low. When we looked at other parameters, we indeed found some statistically significant signal. This is, for example, the Crohn's disease endoscopic index of severity. So basically, the gastroenterologist is counting the number of ulcers during the colonoscopy. And what you can see here is that in the FMT arm, between week 0, which is the time of the FMT, and week 6 weeks later, you can see that the score is decreasing, while in the sham transplantation group, between week 0 and week 6, there is basically no change in the endoscopic score. Here, this is a systemic marker of inflammation, C-reactive protein. You can see that in the FMT arm, there is basically no change in this parameter between FMT and 6 weeks later. Here on this line, you have 6 patients, while on the in the sham transplantation arm, you can see that already 6 weeks after the sham transplantation, there is an increase of the CRP, suggesting that the disease is already less controlled. So what about microbiota? You can see that after a single FMT, there is an increase in the alpha diversity, the diversity of the microbiota in patients who receive FMT, and of course, no change in the patient who received the sham transplantation. In terms of similarity between index between donor and recipient, you can see that the similarity increase after FMT in the FMT arm, while of course it does not in the sham transplantation arm. And interestingly, we saw that there is some kind of long-term effect with some bacteria that are able to remain in the gut for several weeks and even several months. Here we looked, and actually it was the primary endpoint of the study, we looked at the similarity between donor and recipient at week 6. And this was done using 16S sequencing. And what you can see here is that indeed there is no statistically significant difference at this specific time point in terms of similarity between donor and recipient. And actually, we were very disappointed of this result initially. But as we saw some clinical signal, we dig deeper into the data, and we realized that here there was 2 patients that basically were not colonized at all by the microbiota of the donor. We can consider it as a kind of FMT failure. And of course, as this is again a small study with the aim of generating hypotheses, we put them aside. We separated these 2 patients from the others. And then here you can see that there is indeed some effect of FMT in the remaining patients. What is particularly interesting is to know what is happening to these 2 types of patients after the fecal microbiota transplantation. What you can see is that indeed the patient in red with this kind of FMT failure basically behave like the sham transplantation group, and you can see that they relapse quite rapidly. While the patients in this green situation who had a kind of significant engraftment by the donor microbiota have a much better outcome with a very low flare rate. So in this particular setting, it suggests that colonization by donor microbiota is associated with maintenance of remission. What it means is that also we might need to bring this kind of microbiota signal, microbiota treatment regularly and not only once to achieve the maintenance of remission. So now I'm moving to the other topic, which is immune checkpoint inhibitor. We know that the activation of CTLA-4 or PD-1 inactivates T-cell response to cancer, and this is an important mechanism by which the tumor escapes the immune system. So there have been the development of these very interesting drugs that are able to blockade CTLA-4 and PD-1, and this leads to the reactivation of the T-cell response to the cancer. So, of course, as the gut microbiota plays a key role to shape the immune system and the immune response, very rapidly people asked whether this gut microbiota might play a role or might impact the response to this immune checkpoint inhibitor. So this is a study in mice performed by Laurent Zitvogel group in France. And what has been shown here is that basically antibiotics compromise the efficacy of PD-1 blockade in this animal model. Then what is it in human? In this study, pooling patients with different types of cancer, non-small cell lung cancer, renal cell carcinoma, and urothelial carcinoma, again, antibiotic treatment was associated with a shorter progression-free survival and a shorter overall survival. I will not go into the detail into all the other studies that have been performed, but basically a positive role of a high diversity of the gut microbiota and also of specific bacteria, including Faecalibacterium progenitii and Akkermansia mucinifila, was identified. So here are a few of these studies in which you can clearly see that patients with a high diversity of the microbiota or a higher level of Faecalibacterium progenitii had a better response to immune checkpoint inhibitor in the context of metastatic melanoma. So the next question is to know what we can do for these patients who are not responding to this type of treatment. And of course, the idea is to change their microbiota. And there have been two very nice studies published a few months ago in Science evaluating this strategy. These studies are relatively small, again, 10 patients for the first one and 15 patients for the second. They are open label, so they are not controlled. But they include patients with immunotherapy refractory melanoma. And these patients were treated with FMT using microbiota from responders. And basically what this study showed is that in a subset of patients, there is indeed a clinical benefit to do so. In the first study, there was a clinical response in 3 out of 10 patients with potentially a donor effect here. And in the second study, there was a clinical benefit in 6 out of 15 patients. Of course, now we need larger and controlled study, but these results are really encouraging. So I will move now to the second question, which is, can we really standardize fecal microbiota transplantation? So currently, this is the way we are preparing FMT. And as you are going to see, the process is mostly a negative selection process to remove feces at risk of transmitting disease. So first, we see a donor, and there is a medical consultation and a safety checklist. And basically, the process can stop very rapidly. And in most of the cases, actually, this is what is happening. The donor cannot go on with the selection process. If this first step is passed, then there is a very large and extensive biological screening, both in the blood and in the stool. And if these tests are fine, then it opens a kind of window of donation, a donation window in which the donor can give as many stools as he wants. And all these tools are prepared and put in quarantine. At the end of this window, often it's 4 weeks. It can be a little bit longer. It can go to 8 weeks. There is another biological testing, which is basically the same as the first one, so very large, very extensive. And here, based on the results, if there is any abnormality, all these tools here are basically removed, they are destroyed. And it can be painful in some cases because you can basically work for nothing. If the screening, the second screening is fine, then these fecal preparation are moved to the active bank, and then we can use it for fecal microbiota transplantation. Then the next question is, can we do better than that? Can we really standardize the tool? From my point of view, currently, the answer is no. It's basically impossible because we are not able to fully characterize a human fecal sample. I will just give you a few examples. 20 to 50% of shotgun metagenomic sequencing reads cannot be mapped. 30 to 50% of identified genes are of unknown function. And when you move to metabolomics, 80 to 90% of the metabolites cannot be identified. You don't know what are these metabolites. And this is even not mentioning the virus, the archaea, the fungi, the protists, and even the human cells materials that you can find in the stools. So in conclusion, the only indication currently in clinical practice of FMT is recurrent C. difficile infection. They are accumulating data showing efficacy signal in IBD, but still the clear indication and the procedure to use in this patient remain to be determined. All other indications remain in the research field until now. We need randomized control studies, and we also need to define indication and procedures better. I think it is not currently conceivable to fully characterize a human fecal sample. Of course, it is feasible to select specific donors based on the amount of specific components, specific microorganism, richness, or metabolites. But even this strategy cannot be used as a standardization procedure as all the other components are not controlled. What are the perspectives for FMT? The first problem we have, at least in several European countries, is that FMT is considered as a drug, while obviously it does not have the property of a drug. So we probably need a very specific status for FMT, acknowledging the specificity of this product. If we want to move to really standardized treatments based on the microbiome, we need, I think, to build the drug from defined and controlled components. Finally, we have to remember that not everything is about the donor. The recipient is also a major actor in the efficacy of FMT. The recipient immune system notably may have major effects on donor microbiota composition and function, with the effect of the host genetics, antimicrobial peptides, electron acceptors, and immunoglobulin, and particularly IgA. And targeting the recipient might be needed to optimize FMT efficacy. It can be by optimizing the donor-recipient matching, by preparing the gut environment, by targeting the immune system in the recipient, and by controlling the recipient environment, notably the diet. Thank you very much.
Thank you. Thank you, Ari, for this talk. I feel that we have a couple of questions, and we have 1 minute left for it, and the others, I guess we can use pass in during the next sessions. Yeah.
Okay, great. Let's get to a couple questions. The first is from Dr. McFarland in the US. Knowing this is a difficult question, but this is a recipient effect question, Dr. Selkel. Do you have any ideas why some of the FMT has colonization failures? Is there something about the recipients that is different from those that successfully colonize?
Yes, obviously, this is something happening in the recipient. In the study I mentioned, we first look at the colon preparation, because one obvious possibility is that there is the preparation of the colon was not perfect, and indeed it was not the case. The preparation was really the best as we can, very clean. What we noticed is that also these patients were in clinical remission. Of course, clinical remission is based on a threshold. And actually, these two patients were just below this threshold. And when we looked also at the microbiota composition of these patients, they had obviously sign of stronger dysbiosis. And so one of the hypotheses is that probably their intestinal inflammation was still activated. And with a single FMT, as it was done in this study, it was probably not enough to achieve the therapeutic effect.
Okay, thank you. And we have time for one more question. This is from Dr. Vargo in the US. So how much focus has there been on capturing diet and other metadata in both the donors and the recipients?
So until now, I've not seen a lot of studies about that, but I'm aware of several studies trying to combine a dietary intervention on top of the FMT. And this is clearly in the way I was mentioning on the last slide. If we want to maintain, to expand the effect of the FMT, we also have to change the gut environment of the recipient, and obviously diet is a major actor here.
And so this also goes along with the idea there's some symbiotic formulations for these defined communities that are being transplanted, and that I think will be an important development in the future. Cyril, I think we need to move on.
Yeah, we're going to be in trouble otherwise. Yeah, you're right. I'm just going to introduce our next speaker, and this is Hervé Affagar. And the title of his talk is Fecal Microbiota Transfer from Bench to Bedside. So Hervé is an entrepreneur specialized in the healthcare sector. He founded Mad Pharma in late 2014, together with the oncohematologist, Professor Mohamed Mothi, and the great microbiologist, very well known in the field of microbiota, Dr. Joël Doré, after a professional career that spanned multiple industries. Hervé has led Mad Pharma development from its early concept in 2013, and has been at the forefront of the development of the microbiome healthcare ecosystem in France and in Europe. He's also super important in our area here around Lyon and the region. He plays a great role. Today, Mad Pharma is a patient-centric, oncology-focused microbiome company with two full ecosystems, live biotherapeutic programs in clinical development, including over 100 patients treated to date in 3 indications, a total of 45 million euros raised in dilutive and non-dilutive financing, relying on diverse, committed 35-people team with expertise ranging from AI to microbiology, immunology, and oncology. Hervé is also a board member for the French Bioindustry Associations, France Biotech, and an active contributor to Alliance Promotions Microbiota, an organization recently founded to support the microbiome sector development in France. Hervé, thank you for accepting our invitation. It's a great honor, and the floor is yours.
Thank you very much, Cyril, for your introduction. So let me share my screen. Wait a minute. Can somebody confirm that's okay?
Yeah, it's all good. Yeah.
Okay, thank you very much. So thank you very much to the organizers for the invitation. Good morning for those who are in the US. Good afternoon for the others. I'm glad to present today MadPharma, which stands for Microbiota as a Therapy. This is a company indeed that I've founded back in 2014. What we do really at MadPharma, our vision is to create a leader in terms of the microbiome companies, and our objective is really to be the leader in establishing the appropriate ecosystem in patients, so that the patient can either improve the compliance with existing therapies, like it is for immunotherapy, for example, or treat directly certain disease, as you will see from the clinical data I will share with you on GVHD, for example. So that's really our target, and we want to establish the ad hoc signature, I would say, on the patient, the ad hoc ecosystem in the patient's gut. It has been discussed already before, but there's two ways to consider the FMT approach. Our approach is a commercial scale fecal microbiota transfer, so that we can provide a product to the biggest number of patients and doctors, of course. We are not the only one. There's, in order of creation, Reproducibility has been created in the US a while ago, and their objective is to treat Clostridium difficile. They have completed a phase 3 trial already. They are expanding the number of patients treated with their product as part of an open label clinical trial. And we are expecting them to provide the first commercial solution to treat Clostridium difficile with their product, which is named RBX 2660 soon, which means that there will be a commercial solution for physicians. The second, in terms of the date of creation, is MadPharma. We are focusing in a different therapeutic area. Here we are targeting the graft versus host disease, which is basically hematological malignancies, patients receiving stem cell transplantation to cure their disease. And unfortunately, 25% of them will develop a steroid refractory acute GVHD. That's the patient who will be targeting, as long as they have a GI involvement. We have already completed a phase 2 trial, and I'll be sharing those data with you today. And we are expecting to launch our registration trial early next year or by the end of the year. And our product is already available through a specific program here in France, which we call ATU, or an early access program. So the product is also available. And there's another company in the US, Finch, which is developing a product, and they have already completed the first pivotal trial, and they are en route to a second pivotal trial before the end of the year for the start. And here again, that will be another solution for the physicians. And the difference between the one of Reproducibility and the one from Finch, Reproducibility, it's an enema, as it is for MadPharma, and for Finch, this is a capsule. So which means that there will be products available, and those products, they have all followed the drug development framework, because in France, in the US, as in other countries like in the UK or Germany, for example, this FMT product, they are considered as a drug. It has been already discussed a lot by the previous participants, so I won't go into the details, but just for those who just joined the conference right now, here we are talking about some disease which are Clostridium difficile, which is really the key indication, as it has been very clearly presented by Harry just before. But there's also other indications that are of interest regarding the potential of FMT. Multi-drug resistant bacteria is one, ulcerative colitis is one, IABS is one, acute GVHD, autism, and also the restoration of the sensitivity to ICI. So I won't comment on that one because we have some prominent doctors on the conference today. Today, the practice has been led mainly by investigator-initiated trial, which is very good because it has helped to generate a lot of initial proof of concept, I would say. Unfortunately, in the absence of standardization, it's very difficult to pull the data together. So today, each clinical trial is coming with his own conclusion, but it's still difficult to understand beyond Clostridium difficile exactly the potential of FMT because of the lack of standardization. On our side, we are what we are doing is we are developing our own programs. In total, we have already completed 8 clinical trials, and also we are working with academic partners as part of investigator-initiated studies, so that we can also support the effort of generating data, which is very important for the field, but also for us, so that we can better inform and design our product as well as our processes. So the way we have created the company, it's a step-by-step approach. We have been among the first one to treat immunocompromised patients. So in fact, we have treated patients with autologous FMT, with monodonor FMT, with multi-donor FMT, and soon we'll be treating patients also with synthetic FMT. So we have already developed this expertise, and it has started back in 2014 with the first collaboration with INRA, which is mastering the concept of manipulating, storing the microbiome. So that's what we get from them. And we work with them also in 2015 to make it a product that we can administer to human. That has led to the creation of the first GMP platform in Europe to generate enemas for FMT. So that was in September 2016 when we treated the first patient from this platform, from this GMP platform. Then we have developed again a strategic partnership with Bioaster, Institut Gustave Roussy, but also Biocodex, a pharmaceutical company. And Cyril, I've noticed that you want me to say that was a very nice collaboration. So yes, Cyril, it was
exactly.
a nice collaboration. So
thank you.
we got our first clinical results in 2018 with the Odyssey trial, which was in leukemia patients. Here we were doing autologous FMT. We have demonstrated that 2 things. The first one, that there is a very, very strong dysbiosis in those patients receiving chemotherapy and antibiotics. Probably it's very clear today, but back in 2015, it was not so obvious to understand what was the status of the microbiome on those patients. So not only we have demonstrated the strong dysbiosis on those patients, but also we have demonstrated that we can restore their microbiome at 90%, 90, as compared to the baseline at the time of when they have been diagnosed with the acute myeloid leukemia. And once again, it's with the autologous approach. So based on those very encouraging results from, I would say, benefit point of view, but also the safety profile, which was very nice. For example, a month after the FMT, none of the patients treated have developed any infection. So the safety profile was really, really good. So that has encouraged us also to move to the next stage, which is the development of our pooled product, which is allowing the standardization of certain parameters, such as the richness, for example. And we have here started a phase 2 trial in GVHD. And to make a long story short, we have continued with the development of a capsule, because up to 18 we were with enemas. So we have also developed a capsule. We have completed the enrollment of the GVHD patients in 2020. We have initiated our first clinical trial with the capsule still in leukemia with the Simon program. And we have published also interesting data during the ASH, American Society of Hematology, the ASH Congress in December 2020, with a very nice benefit for the patient, including a very nice figure in terms of the overall survival. So as of today, we have treated more than 80 patients, 80 patients in GVHD. So that's pretty nice cohort. So that's the story. Today we have 4 active clinical trials. Active being Heracles, the GVHD clinical trial that we have completed, and we have communicated the top line data a month ago, and now we will be publishing the paper soon on that. There is also the Simon program. This is a capsule program back in acute myeloid leukemia again. We are about to launch another trial in advanced melanoma patients using the MAP13 program, the same as the one on GVHD. And we are preparing for a next clinical trial still in immuno-oncology potentiation with a synthetic program that I will a product that I will develop later on. So the concept of what we are doing, it's really first to restore the richness of the microbiome, because we have seen it's a very important component in terms of getting a clinical benefit for the patients. But not only we want to restore the richness, but also we are able, because of the formulation that we have developed together with INRA, to promote certain commensals, which are very important with regard to the mode of action that we are targeting. So those bacteria, they are mainly butyrate producers, and we are also able to report the fact that this pattern of commensal is always present into the product. So that's another way to look at standardization, where we are always providing this set of butyrate producers so that we can play a certain function, especially with regards to local immune homeostasis and modulation. That's our focus. It has been said before, we could be focusing on many things, but ourselves, we are focusing in oncology, including GVHD. This part is very important, because that's also what I think makes us unique. We have a discovery and data science platform to evaluate our drug candidates, to determine both novel disease and identify biomarkers for microbiome-related conditions. Through this GutPrint platform, we have analyzed already thousands of samples, which enables insight that will pave the way to greater understanding of the impact of microbiome therapy. We do that by getting data on patients per indication. As I said, we have already run 8 clinical trials, the literature data also, but also the healthy donors. Based on the comment that has been made before, by Harry, indeed, it's very hard to find a healthy donor that is fitted to enter into the collection phase to produce a drug. And here we have been screening thousands of donors. We have a huge database regarding the donors, and it's always good to have patients' data when you compare with highly vetted donors too. So that's a very strong asset too. Sorry. So as I said before, it's about biomarker identification, screening and drug candidate assessment, as well as community selection, because our products are always an ecosystem. We are not here proposing to identify one specific element to do the job. Our modality is really focusing on full ecosystem. Then we go through the validation phase, in vitro and in vivo, and then that will become a product candidate, and then we will leverage again our GMP platform, which is able to produce both the native product and the fermented product. So let me check time. So that's the way we are progressing in terms of the development. So that's like a step-by-step approach. Maybe I can skip that one because I've been already talking about that. So going back to the selection process, on the left-hand side, you have the selection process for the native product. So we have screened thousands of donors, and indeed, the attrition rate is very high. And we create pool of 4 to 8 healthy donors. And for example, the standardization of, if you look at richness as a standardization parameter, we are able to report a standard deviation of 3% from one batch to the other. So which is pretty nice for a biological drug. We do 2 products from that, MAT13, the enema, and the other one, 33, which is the capsule. And because we are collecting donors since 5 years now, we have been also able to develop some bank of donors. And we go to the next stage for the future, which is to create the fermented product, which will be based from the donors that we have selected, but also some, I would say, pool of certain donors so that we can obtain the target product profile we are looking for. And those products, they will be mainly MAT34 or MAT3X. It's because, in fact, it really depends on the indication. The basic of the product is to restore the richness, and then we may want to favor, in terms of the abundance, certain bacteria to play a role based on the indication. So that will be 35, 36, and other development. So as I said, that's a little bit some of the visual, but I think you have understand the most important. You see that what I've not mentioned yet is the fact that we have also developed some medical devices going from the administration device and to the collection device. So the collection device, in fact, it allows to protect against oxygen from the collection. So the patient gives his donation into the device and then immediately close it. We can extract the air and it will not open again during the process of production. And then we can produce the different products that are here. Of course, devices, they are CE marked. Everything is done under GMP condition because we are a drug, and it has been patented in terms of the platform, of course. So when we talk about standardization, because indeed it can be a controversial topic. Here, for example, we illustrate, we have several parameters. One is the richness, and you can see here the difference between a monodonor and a multi-donor. So on the monodonor, you will have in average 274 species, OTU species, and for the pool, you're gonna have 455 species. So what does that bring to the patient? We are of the opinion that what will make the difference in terms of the engraftment is really what's going on the patient side. And we feel it's important that we bring the full menu to the patient, and the patient will make his own selection. So to us, it's an advantage to provide, as long as the safety profile is confirmed, it's important to bring the full menu to the patient so that he can make his own selection. So that's what is important for the richness in terms of like doubling the richness. And also you see also the standardization, which is better. You go from 15% to 3% in terms of the variation from one product to the other. So I think this figure is pretty important. GVHD, so our patient, there are those patients with GVHD with GI involvement. You can see from the picture here what that means. And richness is very important, that papers that have been communicated by other groups. Here, maybe I'll skip it. Our data are showing that we have treated more than 80 patients. It's moving very fast. 40 of them, they are ATU patients, early access program patients. And then we have treated 24 patients as part of the phase 2 clinical trial. I won't go into the detail too much, but the results are showing that there's an improvement for patients, including complete response. So those patients, they are literally dying from their GVHD. This is a very severe condition. And those patients, they have been already refractory to steroids. And we have seen here that we can compete and be as good and sometimes even better if you compare to other immunosuppressant drugs. And this is a one-shot treatment because you have only 3 administration and then it's done. You don't treat the patient again. And we see the survival of the patient with a complete response after the survival after a year, which is very nice too for those complete response patients. So those results, they are very encouraging. And they are also confirmed by our early access program data, which are showing an even better results. But of course, it's also because it's a different population. But the survival is very nice too. I see CL showing up, so maybe that means I need to speed up. So this, I have a narrative on the immunotherapy, but it has been discussed before. And coming back to the fermented products, so today, and that's a technology we have partially developed with Bioaster. We leverage on our capacities to develop a full ecosystem fermentation technology. And the challenge here, it's really to grow the bacteria together. And we are able to make it at a certain scale today, which is already enough to consider entering a phase 1 in immuno-oncology. And maybe that's the summary slide, and I will let it here so that I can take questions, Cyril.
Okay, thank you, Hervé.
Okay, Hervé, we have several questions, and some others came in on the panel as well, but I think those are more in-depth questions, and we'll wait for the full panel discussion. but I'll tackle some of these more direct questions now, or actually Hervé will. Are you concerned about COVID virus, and are you screening for that in the feces of your donors?
Yeah, absolutely. To make it very short, everything has been suspended in France, where we are collected in March 2020, because we didn't know exactly what would happen. We know that there's a SARS-CoV found in feces, and we had to develop a test. We did that together with the group Harry mentioned before, the Groupe Français de Transplantation Fécale. And in November 2020, we have been able to restart based on the use of those kind of tests that we have developed. We have been to reuse both collection and administration in patients. And I think France is one of the countries which has restarted the first, because, for example, in the US, they have restarted in February 21. So yes, we were concerned. Yes, we had to adapt ourselves and our streaming strategy. And we did that like within a 6-month period.
And I think this topic maybe deserves a little more expansion on in the panel discussion, because what other specific pathogens need to be screened for, and how much does that need to be done? Another question is, how are you evaluating the bacterial abundance, and what DNA kit are you using for fecal sample extraction?
Yeah, let's keep it for the panel discussion so that I can get the answer before.
Okay, and I think we have time for just one more. Do you have any concerns about donor selection process introducing a bias or excluding important sources of diversity in phenotypes from the donor bank? So it looks like you're pooling donor material, which should address some of that diversity issue.
Yeah. That, I mean, because of the pooling that comes with advantage and disadvantage, but one advantage is the fact that you are like making an average donor. You have a donor with everything in it. So as long as it is safe, it's okay, and you are stepping back from this kind of issue with the strategy that we have developed.
Thank you. We have one other in the Q&A box, and I guess I should ask this. What is your opinion on the hypothesis that it's actually bacterial phage that are the antigenic mimemic for tumor-associated antigens? So there was an interesting paper on that a couple of years ago, and how would we integrate this information into the FMT approach?
Yeah, as it has been said by Chris in the first presentation, we have not looked that much into the phage at this stage. But as we are getting closer and closer to the market, that's absolutely a question that we're going to address too. But today we can't report data on that specific question. We are working on the phage analysis, but we do not have data yet. But of course, we need to be able to answer that question at one stage.
Thank you so much. We will get to the other questions during panel discussion.
Thank you very much to the attendees.
Thank you, Hervé. We're going to move to the next speaker, and the next one is Vincent Thomas, Dr. Vincent Thomas. Vincent works for Bioaster. He's not in Lyon. He's located in our labs in Paris on the Pasteur Institute campus. Vincent holds a PharmD, and he has a PhD in microbial ecology. He's led his career at the interface of different public and private structure on various R&D topics. His thesis and postdoctorate project were carried out in partnership with Suez Group in France, and focused on the study of interactions between free-living amoeba, in the environment and in different types of microorganisms, including Legionella, new bacterial species related to chlamydia and mycobacteria, and also giant viruses. He then joined the US company Steris to manage more than than 6 years, doing more than 6 years, an R&D lab located at the CEA, the Centre d'Énergie Atomique, in Fontenay-aux-Roses, close to Paris, with an activity mainly oriented towards the control of infectious risk with respect to classical and emerging microorganisms, as well as Brian. In 2012, he was hired by a great jewel also of the microbiome field, that is, and this, in a startup named Enterome, a leader in the field, notably to work in collaboration with INRAE and Metagenopolis on the implementation validation of an integrated metagenomic analysis pipeline for fecal samples. And since 2015 is with us at Bioaster, where he's in charge of positioning the Institute's strategy and vision on the field of microbiome research, and he's done a pretty good job until now, defining the key technological development for Bioaster research in the field, he's working, he's set up a lot of collaboration within our institute between private companies and great academics in France and also abroad. He published more than 40 peer-reviewed papers dealing in the various topics he addressed during his career. And the title of his talk right now is New Strategy for Stratifications and Longitudinal Monitoring of Microbiota. Thank you, Vincent, for accepting to be part of this speaker panel today.
Thank you, Cyril, for the kind introduction. Can you see the slides? Yeah.
Okay. Yeah, they're on.
Thanks.
Maybe you could switch to presentation mode because you're on the normal mode.
Is it? I would prefer to stay because I have two screens, so maybe.
Okay.
For you, yeah. So hello everybody. Yeah. So my talk, I will give a talk about as Cyril mentioned already, about the new strategies for the stratification and the longitudinal monitoring of microbiota. So this can be for gut microbiota, but also for other kind of microbiota. So the context, as all of you know, is that there's really a lot of interest. There's been a lot of interest these last few years about the gut microbiome, but also other microbiomes. But on the gut microbiome, it's been associated with many diseases and very important diseases such as cancer, as it's been said already, but also metabolic syndrome and IBD, as already mentioned. And that the number of potential applications is huge, with, for instance, the potential to stratify patients based on their gut microbiome compositions, the potential to develop new diagnostics products, new treatments. Also, it can be used for drug discovery. So really, there's a huge interest, but there are also still caveats, and it's been mentioned in the first talk. For instance, the gut microbiome analysis is mainly performed today using sequencing technologies, and these technologies, they still remain time-consuming, and often they give a huge amount of data and results that might be difficult to interpret. And as it's been mentioned, there are a lot of technical biases that can be an issue using these technologies. So the conclusion is that there's a strong need for additional technologies that can measure important parameters that have been linked to health status in a variety of diseases. So this can include, for instance, parameters related to the overall microbiota, such as richness, diversity, bacterial load, so meaning the bacterial numbers per gram of fecal matter, but also the bacterial viability and the proportions of gram-positive versus gram-negative bacteria.
Sorry, Vincent, we're still on your first slide right now. Are you switching your slide?
Yeah, I switched over.
Okay. Yeah, I think you're on a presentation mode on the full screen on your second screen, but this is not the screen we can see right now. Yeah.
Okay, I will try to adjust.
Yeah, this is all good. Yeah.
It's okay?
Yeah.
Sorry. Okay.
Yeah.
So that's the text I was just explaining. And so it can also be of interest to be able to detect specific bacterial species of interest that have been linked to a good or a poor prognosis. But there can also be an interest in detecting not only bacteria, but also yeasts and fungi and phages that it's been mentioned. And I think a major point is that longitudinal analysis is still a strong and relatively unmet need. So that's the context. And now the focus, I will focus on the gut microbiome, the monitoring of gut microbiome composition in the context of immune checkpoint inhibitor therapies. With this question, should gut microbiome composition be taken into account during these treatments? So it's been in this figure, which is it's been published by the team of Dr. Vargo in 2019. In this summary figure, the key message is that the healthy microbiome, and it's been mentioned already, patients with a healthy microbiome are more likely to respond to the treatment. So it's been demonstrated in a few important papers these last few years that have been published in really major journals. And on the other side, patients present with a dysbiotic microbiome, so meaning a low diversity of microbiota, for instance, overwhelming inflammatory. These patients, and this can also be potentially due to the previous or the concomitant treatments with other drugs, such as antibiotics or proton pump inhibitors, that can affect the gut microbiome compositions. These patients, they are less likely to respond to the treatment. So that's a very important point to consider. And it's been, as Ari already mentioned, there's been a couple of companion papers published recently in Science that very nicely demonstrate that FMT can be an option that needs to be explored in this context, because it seems like it could restore the response to the treatment in refractory patients. So I will not go into the details because these are very well-known papers that have been published. So this being said, one can understand that there's really a need, or at least there's a need to explore if it's important to monitor gut microbiome composition at various stages of these kind of treatments. So this can be before starting the treatment, to kind of stratify the patient, to select the patients that are more likely to respond to the treatment versus those that are likely to not respond to the treatment. And during the therapy, it can be also very important, because if there's due to, for instance, to additional treatments or drugs, if there's an impact on the microbiome composition, it has to be monitored. And for obviously, during the course of FMT, if FMT is an option, it can be very important to be able to monitor engraftment of the microbiome in the patient, and the long-term effects of the FMT process. So how durable is the engraftment, and what microbes or functional phenotypes are associated with the response. So what we propose, and what we've put a lot of effort on these last few years in Bioaster, is to investigate flow cytometry as a tool for fecal microbiome monitoring. Now, I will give you a few examples of what we've done and what can be done in this field. So this was, for instance, a preliminary kind of proof of concept study conducted with the company, the preclinical model company Synbios, which is based in France, Lyon. So this was done on non-human primates, and this was a very simple study. It only consisted in giving antibiotics to the animals and collecting fecal samples before the treatment, during the treatment, and after the treatment. So these were quite drastic antibiotic treatments. And the fecal samples were analyzed using flow cytometry and sequencing. So these are the results that we obtained using flow cytometry. So we use different staining methods for the fecal samples. And as you can see, we can really easily detect changes in the gut microbiome composition using flow cytometry during the course of the antibiotic treatment. If you compare the dot plots of the animals before the treatment and during the treatment, you can really see major differences in the dot plots, and you can also see that the follow-up after the treatment was quite short, and it seems like the microbiome was not able to recover, and it can be seen on the dot plot too. And now, for some of these samples, not all of them, but for some of them, we also did the sequencing analysis. So these are the ones that were used also for sequencing. And these are the results of the sequencing. So what you can see is that there's a kind of inter-individual and intra-individual variability in the fecal microbiome composition. And you can also see, obviously, that there's a very strong impact of the antibiotic treatment on the fecal microbiome composition. Since after 3 days of treatment, you can detect mostly lactobacilli in the animals that received the drugs. And then after the treatment has been stopped for 3 days, there's a recovery, but with specific, it seems like specific groups or genera of bacteria appear first, including Fecalibacterium, which was a bit surprising. And then you can also see that even after 5 days after the end of the treatment, it looks like the gut microbiome is still different from what it was before the treatment. So this is with the sequencing, and this is the classical way of doing analyzing the data using the relative abundancy profiling. But what you can do also when you do flow cytometry, and it's been published in this nice paper by Dr. Van de Putte in 2017, you can take into account the bacterial load in the fecal sample to correct the sequencing results. So it gives a completely different picture. And you can see that even if the lactobacilli was the most prevalent species in the fecal samples after the treatment, they are in very low numbers compared to the microbiome, the full microbiome before the treatment. And then you can, and this is ongoing now, this is something that we are investigating, you can try to link the results you observe using sequencing with the results you observe with the flow cytometry, because it seems like, specific groups of bacteria that appear and that you can detect using flow cytometry, you can also detect using sequencing. So the goal now, and what we are trying to achieve, is to kind of generate some kind of maps of the microbiome using flow cytometry, and try to predict to which groups or genera of bacteria will specific subgroups that we detect in flow cytometry, to which groups they correspond by doing the parallel analysis using sequencing. So this is ongoing. Another way of using flow cytometry for the gut microbiome analysis is to measure bacterial variability. So this is the very classical live dead staining method that has been used for years in many kind of applications. But here we were lucky enough to have a specific device and technology allowing us to do all the analysis and sorting experiments under strictly anaerobic conditions. So we were very interested in trying to sort and cultivate live and dead bacteria, and then try to see if there's really what's the difference between the number of bacteria that can be cultivated from the live versus from the dead fractions. And we've done this with a number of commensal species. So this is still a limited number, but at least we've tried with several representative species. And what we can see is that there's around 2 to 3 log differences between the bacterial numbers that can be cultivated from the dead and from the live fractions. So it seems like the live dead staining, at least for this limited number of species, is appropriate to evaluate the bacterial viability in the stool samples. This has been done as a collaborative work with Matt Pharma during a PhD thesis. And during this work, we also evaluated the gram staining because it's been also really reported that dysbiosis is often linked to a more abundant population of gram-negative bacteria. So it can be of interest, especially in longitudinal studies, to monitor the proportions of gram-negative versus gram-positive bacteria. So we were very interested in doing this, and we tested different staining methods. And the one that gave the best results was a combination of the lectin wet germ agglutinin with vancomycin body pile. In this case, if you use these two stains together, you can really correlate the number of bacteria that were detected as gram-positive using flow cytometry with the number of gram bacteria that were classified as gram-positive from the 16S sequencing results. So this is very encouraging because it means that you can you have a good instrument to measure the gram-positive bacteria in the microbiome. Another tool that we are developing now, we put a lot of effort on it, is to develop antibodies that target specific bacterial species of interest in the gut microbiome. So we developed a collection of antibodies directed against, for instance, Akkermansia multinifila. Ari mentioned that it's been associated with an enhanced response to immune checkpoint inhibitors, but it has also demonstrated to have beneficial properties in the case of insulin resistance and glucose tolerance. We developed also antibodies directed against Faecalibacterium prosnidii that belong to different phylogroups, because what is called now Faecalibacterium prosnidii is quite a huge group of bacteria. So it's interesting to have antibodies that can differentiate between these phylogroups. And so we have a collection of antibodies, and I will skip this one because this is just to mention that Faecalibacterium has been associated with a good response to immune checkpoint blockers in this paper. And this is the one for Akkermansia multinifila that has also been associated with the good response to the treatment. What we've done, for instance, and this is an example with the antibodies directed against Faecalibacterium prosnidii phylogroups, we mixed fecal samples from healthy volunteers, and that they were hybridized with the antibodies, and then we sorted the fractions that were stained with the antibodies, and then we did the 16S sequencing. So here you can see that in the raw material there was less than 5% of the sequences that were affiliated to Faecalibacterium, whereas in the sorted fraction you get as much as around 95% of the fraction of the sequences that are affiliated to the Faecalibacterium for one antibody, the one which is directed against the phylogroup 1. And this is around 80% for the one, sorry, that is directed against the phylogroup 2. Sorry. And interestingly, it seems like these antibodies, they still recognize different strains or categories of strains that belong to the same phylogroup. So this is under submission. And what can be done once you have these antibodies and that they've been validated is that you can use them really to analyze the target bacteria in the fecal samples. And this is an example with fecal samples that were collected from 5 healthy volunteers. You can gate, for instance, the live bacteria. And then you can analyze among the live bacteria the ones that are stained with the antibodies. And so we've seen some huge differences from one volunteer to another with proportions that range from 0.1% to almost 19% of the live bacteria that are stained by the antibody. So this is the conclusion. So we've validated and we've developed, and we are still developing a series of staining methods that are compatible with the flow cytometric analysis to explore fecal microbiota composition at various levels of precisions. So we take into account at this stage the bacterial load, proportions of live versus dead bacteria, the gram-positive versus gram-negative, the bacterial population complexity that can be detected or estimated based on simple physical parameters, but also based on DNA staining. And we also have specific antibodies that are directed against commensal species of interest. The ultimate goal is to generate this kind of analysis that corresponds to multi-parametric flow cytometry data analysis, and based on this, to be able to predict which patients is more likely to be a good or bad responder, or to have a low or high risk of toxicity due to drug interaction with the microbiome. So really to take into account the microbiome during the course of the treatment. And that's it. And thank you for your attention.
Thank you, Vincent. Phil, did you see questions? Did you receive questions on your side for Vincent, or we're slightly behind schedule? So
we have no new questions in the Q&A box, so I think we can go ahead to Stephanie introducing the panel.
Yeah, I'm going to introduce Stephanie quickly because Vincent is out of breath, and I'm going to take the relay on introducing Stephanie. So Stephanie joined the group of this webinar while we were constructing it, and she was a great asset, I think, for. Oops, okay, I just received a message from Zoom that I need to restart my video. Oh, can you? Oops, so yeah, sorry, my video was not on, so I got caught by the police. Sorry. So Stephanie is Servetas, I hope I said it well, is a microbiologist in the complex microbial systems group at the National Institute of Standards and Technology, the NIST. And Stephanie's research is focused on whole cell and multi-omic measurement of microbial communities, both natural and contrived. While at NIST, she's worked with on the development of reference material for pathogen detection, which includes leading the development of viable bacterial mixture containing over 50 species to validate new pathogens detection platforms. And most recently, she got into effort to develop a whole cells tool gut microbiome reference material. And this material is now being characterized using multi-omic approach with the primary goal of supporting metabolomics and metagenomics biomarker discovery and characterizations. So she's arrived at NIST in 2018 after completing her PhD in emerging infectious disease at the Uniform Services University in Bethesda in Maryland. And she'll be leading the panel discussions. Thank you, Stephanie.
Thank you for that introduction. So as Cyril said, we're going to move on to the panel discussion portion of the meeting. And just as a reminder, we'll continue to monitor the Q&A box for questions from the audience. So please continue to write those in. And if you would like your question to be directed to a specific panel member, please include their name. So in addition to our speakers, Dr. Jennifer Vargo and Dr. Gary Wu will be joining us for the discussion session. Dr. Vargo is Professor of Surgical Oncology and Genomic Medicine at the University of Texas MD Anderson Cancer Center. She is also the founder and leader of the Program for Innovative Microbiome and Translational Research, or PRIME-TR. Dr. Vargo is recognized as a world leader in research on the impact of tumor and gut microbiome in patients with cancer. She has demonstrated that microbes within the gut can shape responses to treatment with immune checkpoint blockade, and recently showed modulation of the gut microbiome can overcome resistance to this type of therapy in patients. One of Dr. Vargo's recent publications looked at the ability of FMTs to augment immunotherapy in patients with melanoma or other cancers. Dr. Wu is the Ferdinand G. Weisbrod Professor in Gastroenterology at the University of Pennsylvania Perelman School of Medicine. He is the director of the Penn Center for Nutritional Science and Medicine, the co-director of the Penn CHOP Microbiome Program, and the co-director of the NIH-funded Center for Molecular Studies of Digestive and Liver Disease. Research in the Wu Lab currently focuses on the impact of diet on the gut microbiome and its relationship to several aspects of post-metabolism, including nitrogen balance, intestinal oxygen regulation, and epithelial intermediary metabolism. Dr. Wu recently published an article examining the FMT effectiveness in real-world practices based on the FMT National Registry. So I would like to welcome our two new panel members and direct our first question at Dr. Vargo and Dr. Wu. So one of the first steps in the FMT process, and it's a very important step and has been mentioned by several of our speakers, so I'm sure this will be a very lively starting point, is donor selection. So is there a way to standardize this selection so that you can ensure an optimal donor with respect to both safety and efficacy? And along with those lines, when we think about standardization, is that going to be some type of criteria that are outlined? And I think one thing that comes up a lot is the word healthy. You pick a healthy donor, but how do we define a healthy donor? So with that, Dr. Vargo, why don't we start with you?
Great. Thanks very much, Stephanie. And I'd first like to thank the organizers for the invitation to participate, and thank you to the speakers, the participants, and all for your important work in the space. Our patients need you, and this is definitely, I think this has tremendous traction to make a huge difference, not only in cancer and other diseases, but in overall precision health, modulating gut microbes. And so, the question again is there a way to standardize the selection to ensure optimal donor with regard to safety and efficacy? Are there specific criteria that could be standardized, and how do we start to define those? So I think in answer to those questions, just reflecting back on Dr. Sokol's talk, there's a lot of dark matter, if you will, when it comes to what we know about FMT. But nonetheless, it's really the most powerful tool to date that we have to modulate the gut microbiome. So I think, it's here to stay, at least, in the short term, until we develop better next generation therapeutics. But I do think we need to augment what we know through improving reference databases, including analysis of phage, and phage has been brought up quite a bit. And we also need to take into account the impact of diet, medications, and other variables. And certainly Gary can speak to that a bit. We've actually looked in our own immunotherapy treated cohorts, and we find that if patients have a high fiber diet, they actually have better outcomes to treatment. And so taking fiber and other variables within the diet into account with donors as well as recipients, I think is really critical. And interestingly, in that trial from Israel that was published in Science, they actually advocated a Mediterranean diet to their donors. So, so a little food for thought, if you will, there. But, we do have to start somewhere. So with regard to safety and efficacy, is there an ideal donor that would not only enhance, when I speak from the cancer space, enhance response to immune checkpoint blockade and also reduce toxicity? And I think it's a great question, because a lot of people posit that, well, hey, you want to actually, isn't enhancing response to immunotherapy kind of, you want to improve or you want to augment inflammation. And it's actually quite the opposite. And what we find is that people have actually looked at this, and by having a more favorable gut microbiota with higher diversity, more of the response-associated taxa, not only do you improve response, but you also reduce the likelihood of toxicity. And Georgina Long and others have shown that in neoadjuvant studies. And so I think you can kind of find that sweet spot where, like having the right microbiome, what's the right microbiome, of course, is the critical question, but will not only improve response, but also will reduce toxicity. So how do we get there? So I think taxonomy is one way, and we've certainly looked at taxonomy as a possible way of, really trying to engineer the right consortia or augment, even use it as a prognostic biomarker, looking at rumino or things like that, as far as profiling patients to see whether or not they're going to respond, and then selecting donors based on an abundance of rumino, et cetera. But taxonomy is limited, right? And so looking at functional aspects of the microbiota is also critically important. Is there a common denominator with regard to function? And we and others are certainly looking at that. And then, I think folding in diet and other variables is really important. And then, like I was really intrigued by Vincent's talk on flow cytometry. Can we bring in novel aspects of looking at different microbiota using novel approaches too? So kind of a long-winded way of saying, I think, yes, there are ways to standardize the selection to really increase efficacy and reduce toxicity and to improve safety. The criteria, we have some basic criteria, but we can iterate on that and do much better. And then I think as a group, through getting together, through consortia like this is really the best way of starting to define those criteria and standardizing them.
So I'll just add a couple of brief comments. I largely agree with what Jennifer is saying. First of all, I too want to thank everybody for inviting me to participate in this. It's really a wonderful opportunity to hear about really great science, but then to explore both the opportunities and the challenges in the area. I think that, if somebody were to ask me about FMT, and I actually participated in some of the initial meetings with the FDA in the United States in 2013, when FMT was just getting started and people were thinking about regulation. And at that time, people were incredibly worried about pathogen transmission. Some really bad things are that could happen with FMT. Now, I'm not saying that it's completely safe, that in fact there have been some rare incidents of pathogen transmission. But I think as proof of concept, at least in the short and intermediate term, it seems to be relatively well accepted that there aren't a lot of huge warning signals right now. And I think that that is actually encouraging, that you can take a very complex consortium, pretty much undefined, put into another individual, and at least in the short and intermediate term, not have some big warning signal. As was Stephanie mentioned, we do have an FMT National Registry to sort of prospectively monitor individuals. The cohort is relatively modest, but in a prospective study, we've not actually seen any significant alteration. So I think that that's promising. So that's from the standpoint of trying to transmit something bad. But then the other question is, so how do you make predictions on what is actually going to be good? And I think that our speakers have highlighted many areas that we don't currently understand that could be improved upon. the issue is really technology, and there are just some very fundamental questions that still remain in the field. I think a lot about when we first started to do our sequencing over a decade ago here in some of our samples, we actually were wondering about the heterogeneity of stool. Like even if you treat somebody with something, when you collect the material and you just take an aliquot off the surface of the stool versus the core of the stool, how often you actually collect it, what is the texture of the stool, how much water content is, all of these things will actually have an impact what you actually see. So we heard a lot of really nice, I think, talks about precision and sensitivity and specificity, but I think a very significant factor in trying to actually define the composition of what you're actually putting into people are things that are currently not very well controlled. And that's where you're actually getting the sample from, and many other factors in the environment of the gut that actually regulate the composition of that material that you're putting into people. The other complex thing is that it's a living consortium, obviously, right? And so it may look a certain way when you put it into somebody, but it's going to change. And so I actually agree with a lot of people that you do have to take into consideration the recipient, even before the engraftment of the organisms or providing material. And so how do you go about doing that? Well, I mean, I think that there are a significant number of studies actually showing that there is a personalized effect in engraftment. And to a certain extent, you can make predictions by what is already there or the treatment of the individual as to what is actually going to engraft. So I sort of favor the notion, and I know it's early days right now, that it's not only the donor composition, but actually knowing what the recipient actually is. And between those two, I think you might have a better understanding of what might engraft. Now, is engraftment important in function? It would sort of make sense that it is. And we can talk a little bit more about what engraftment actually means in terms of function, but I'm just going to pause there, and that's some of my initial comments.
Great. If any of the other panel members would like to comment, please feel free to chime in.
Stephanie, why don't I jump in with a couple of the Q&A box questions, and I'm going to go back to some of them that came in right at the beginning, and then we'll try to work our way through. Can anyone in the panel elaborate on any studies of FMT affecting the aging process? And I'll just leave it open for someone who wants to address any given question. We'll take a couple panel member comments and then try to move on. But it's an interesting question. I don't know of any studies, but perhaps someone on the panel does on FMT affecting aging. And
there is data in mice about some feature associated with aging, such as brain function, these kind of things, but nothing to my knowledge in human, at least.
Yeah, and I know Ashi, we were at Johns Hopkins is studying this with regard to the microbiome and aging and cancer now, actually. So hopefully we'll have some data from Ashi, and we're working with her a little bit on this as well.
I think this covers kind of the area, the spectrum of chronic diseases which go along with aging, and there are a number of indications there.
Yeah, I think that Li Ping Zhao at Rutgers has a lot of interest in this area also. So I do agree with Harry that there are mouse studies that show some cause and effect relationship and associations in humans, but that's where the field is right now.
Okay, the next question coming in. So this is interesting. How can we ensure reproducibility in donor material after co-fermentation of the full ecosystem that I think Harry was talking about? So that was once you start to grow these and expand these communities out, it may affect reproducibility. So I'm wondering if you might comment on that.
Is the question for me?
Yes,
I think so.
Either one.
I think so. No, I think it's for FMT.
You can answer if you want, Ari, but no. Okay, no. Indeed, this was a huge challenge to grow the bacteria together. So we have tested several conditions, and I can't go too much into the details because there's a lot of savoir-faire here, but what I can say, it's of course, it has been very tough to control the growth of certain bacteria that were taking the lead on others, and especially those who you don't want. So you need to find a way to deal with the oxygen, pH regulation, and all of that, so that you have a consistent fermentation. But maybe I can answer on the way we measure the reproducibility, because the process by itself, as I said, it's a little bit of it's a lot of savoir-faire that we are not ready to share yet. But the similarity using indexes like Bray-Curtis, for example, will be very important, so that we can measure along the process. Viability will be very important too, and we are also developing some patterns that are very part of the core microbiome. So we have analyzed the literature and set some patterns on what we would consider the core microbiome, and we make sure that all those parameters, they are kept at the same level if you compare the initial material. But it has taken like 4 years to be able to see the beginning of a result, because it has been very difficult, and we had to mix different conditions. We have also to mix pre-fermentation with other products so that you can delay the oxygen. So that was pretty challenging. It has taken more than 4 years, and today we can produce up to 3 liters of microbiome.
Can I ask Herb a question? Actually, it occurred to me as you were giving your very nice talk. So the alternative approach to actually doing fermentation and sort of designing a complex community would be designing a consortium of organisms that were individually developed in a laboratory setting and then combining them together. So can you comment a little bit about the advantages and disadvantages of both those types of approaches?
Maybe, and there's also people around the table developing monoculture, so maybe it would be great to get their feedback. But ourselves, the reason why we wanted to grow by using co-culture, it's because we think also from an industrial point of view, it's more convenient, because if you were, once again, we start from the beginning, which is our vision. It's really to set ecosystems in patients, and we don't. Maybe at the end of the day, we will end up with 100 species, 250 species. It really depends on what you want to do, but we know that we will not end up with 10 species, for example. And because we have this large community, if you, from an industrial point of view, that will be tough to culture each of them separately and then do an assembly. And also you may lose some, the concept of cooperation between the bacteria, because you don't know what's going to happen when you will mix them together again. But from an industrial point of view, there's also an advantage to have the right mix from the beginning and develop it in a batch way, so that you can produce your lot in one run, as opposed to doing it separately and then doing an assembly. That's our positioning also regarding the industrial scale-up, where we think developing 50 species separately and then doing an assembly, which will be a challenge from an industrial point of view. But I know there's people developing also monostrain development and everything, so maybe they can comment on their approach too.
Thank you for that. There's kind of a follow-on question to that, Gary, and you led into something I was going to ask. But for the entire panel, can you comment on controlling risks from pooled and banked donor material? This, of course, improves diversity and may improve efficacy, but it increases the risk of introduced pathogens and maybe increases the risk of other things as well. So there's a balance to be struck there. And I'm wondering if anyone can comment on how we reach that balance.
maybe I can start. I mean, here we need also to keep in mind, as it has been said by Gary, that it's not like a plasma or something else. Here it's a living microorganism assembly, which is living together. So we are testing pathogen. The screening will be like 45 different kinds of pathogens you're looking for. And you do that on any collection, any donation. You can decide to do it every day. Every time you have a donation, you can select. So that's on the safety aspect. And then when it comes to pooling, we are pooling here 4 to 8 donors maximum. So I would say the risk of having a pathogen which is not detected or by the current technologies, I think is inherent to any donation. But here you are not multiplying the risk like it is for the plasma business, for example, where you are putting together thousands or hundreds of thousands of donations together. So here, that's a different story, because to achieve the richness standardization, you don't need that many donors. So the risk is really limited in terms of the additional risk, and the benefit could be higher because, as we see in the literature, there have been a lot of evidences showing that the diversity and the richness would be really a key differentiator in terms of getting a clinical benefit, of course. And today, as I said, we have treated at MaaT Pharma, we are treating only immunocompromised patients, whether they are acute myeloid leukemia patients or GVHD patients. And today we do not report any safety adverse events that are related to the product in terms of infections. So that's confirmed what we see in Clostridium difficile 2 with this kind of product. But yeah, once again, the additional risk is very limited, and the benefit is, I think, very high.
Maybe I can comment a little bit on that. Of course, I think, and we all know and are convinced that diversity is very important, but I think it's not the same to have the diversity in one ecosystem, which is basically built to work together and to mix different ecosystems. To reach the diversity. So I'm not sure we can really compare these two types of diversity, one ecosystem or pooling different type of ecosystem. Another aspect is that from a research point of view, one of the, I think from my perspective, one of the big interests of doing FMT in a research setting is to try to identify the actor of the therapeutic effect. And it might be metabolites, combination of microorganisms, and so on. And of course, if you pool different donors, it makes it much more difficult to identify these actors.
I would like to answer this, and I've been very fast in my introduction, but I've worked in different industries, not only healthcare industry, and the genealogy concept is a concept which is very well known, and in order, like in the steel business, for example, where you are mixing different kind of steels to get your final composition, and there's tools. And for example, the genealogy of what we are doing is absolutely accurate. Doing shotgun sequencing, we can map up to what was on the donor side, even if we pool, and that's very precise at the end of the day. It's not only about putting something in the recipient and see the clinical benefit. There's all the assays around that, including the fact that we are performing a full genealogy at the gene level. So I will not, maybe it's better, I mean, that's my opinion, I would say. But I think it's something we can overcome with the technology where we are today.
I would say one consideration.
Gary, go ahead.
One consideration, which is largely hypothetical right now, is that if you actually pool donors together, and let's say there was an imbalance in some type of adverse outcome between cases and controls that you actually saw, it might actually be more difficult to try to understand what it was that was leading to an adverse outcome if you pooled the samples together, unless you actually knew what the offender actually was, but you might not. And so the issue is then, it's probably hard enough to find it from one donor, but if you pool donors together, then you have a multifactorial problem where it could be a combination of any one or two or more of those donors, or it could be a single donor out of a number of those donors when you combine it. So again, it's a hypothetical right now, but given the complexity of what we're actually dealing with and people using this as a therapeutic for many different types of diseases, at least exploring it, I think it's something that should be taken into consideration.
Thank you. Jennifer, you want to comment?
Yeah, I'm squirming in my seat here. I think, I love this conversation. And it's like, I think if we reflect on why does diversity matter, right? I think it's an important question. And in my view, diversity matters, not just because you have more microbes, or more diverse microbes, but because there's functional redundancy, so it all boils down. If in at least, my hypothesis would be that if you have a more diverse microbiome, you can fill, holes in the functional repertoire, if you will, of the gut microbiota, that are performing critical functions and roles. And so I love diversity. I think, it matters universally, but, it does, having pooled donors, just approaching it that way does make it complex when you come to develop next-gen therapeutics, and for safety reasons, and I think there have been regulatory bodies, including in the US, that have really been concerned about the prospect of pooling donors because of potential risk. And it does make it harder to boil down, like, what are the contributing factors, that are actually enhancing response, reducing toxicity. And so, though I personally, at least in the short term, love the approach of potentially pooling donors as long as it's safe, I think it does make it a bit more tricky to get to that, next gen therapeutic. But, my idea is it's more of function, and I know there's a question on function, so we'll get there.
And I would say this is a concern, at least in the US, the FDA has about pooled materials. And I would like to hear Chrissy also comment on this, because I think from a regulatory point of view, the more defined we can become, the better it is. There's this idea, certainly diversity is good, but is more necessarily better. And this is a problem for the regulatory agencies to really tackle. But Hervé, I think you first, and then I'd like to hear Chrissy's thoughts on that as well.
Yeah, no problem. And I just want to clarify also our positioning, because we are not here to do academic research. We are here to develop products. And the fact that we could also, we can bring the same product as long as we agree on the parameters for standardization, we can bring the same product to all patients. And I think it's not something we should oppose to the clinical, the academic research, where, I mean, at one stage we will be more defining, that is for sure. But today, when I see that I have a positive clinical benefit on GVHD patients, or patients, they have a survival at 22% at 2 months. So, I mean, let's do it. And then we are developing it as safe as we can. And it doesn't, I think it's not something which is against the fact that we need to be better in terms of defining as a next step. I'm not saying it's one or the other. I think the next step is what we are proposing. And there's also the next step again after, of course.
Right. And it depends on the indication too. I mean, it's like you look at, for C. diff versus GVH versus, and then you get to the more nuanced IBD, cancer, I think, and you have to take the indication into consideration as well, but great points.
And we need the academic to help us to define the signatures that we want for the next generation.
That's right. We're all here for you.
Okay, Chrissy, do you want to quickly comment, or were your thoughts covered by the previous talks?
Well, I quite agree with what everybody said, and I know this is the best approach we have currently, and if it works for some patients, that's what, excuse me, that's what we have. However, I do agree that at some point we need to find how it works and go more specifically, more targeted about it. And then we will be able to better regulate what we give to the patients. But for me, from my perspective, until we are confident on our data and on how we analyze our data and if it's accurate or not, until we have that sorted, I'm not sure how easy we can reach the point to be confident about FMT or therapeutics like that.
So thank you. I'm gonna rephrase this and try to tie this topic up with a little more provocative way to question this, and that is, do we know enough to propose selective culture and expansion processes from donor material, or are we better off forming synthetic consortia from isolated cultures? Because these synthetic consortia have had very limited success so far. So why haven't we been able to better define this? And this is kind of an open-ended question, but I'd like to hear the panelists' thoughts on that.
Personally, I have no religion. I mean, I think both modalities are okay as long as they have demonstrated efficacy in patients, and that's it. I mean, ourselves, we are a big fan of ecosystem approach, and we want to develop a fermented version of the product because of that. And but once again, all modalities based on the indication may have a benefit for the patient, and I mean, we just need clinical trials.
Mm-hmm.
Maybe I can just talk about. We are very much implicated in trying to cultivate bacteria from the gut microbiome, and many of these bugs, they are very difficult to cultivate, and you will spend a lot of effort and a lot of time trying to cultivate and then producing at the industrial level. So these defined consortial products, you will never get as much diversity again as you have in the initial stool samples, and it will still take years before we are able to cultivate all the bugs and then use it in product.
Thank you. Good point.
I think that there are multiple levels at which we need to increase our understanding. I think one component of that is really definitive proof that engraftment is actually important for a particular outcome. There's sort of a hint that that might be the case, and I think that Harriet actually presented really nice data along those lines. But I think we need to understand that a little bit more. And if we do believe that in particular diseases, engraftment is important, then the issue is what type of engraftment are we talking about? Short-term, long-term? What are the organisms? And then the issue is, how do you, let's say you answer those two and they're both in the affirmative, then the issue is, how are you going to develop a product that reliably will engraft in a number of individuals? Right now, it sounds like, at least in the FMT studies with Eric Alm and others, that engraftment tends to be somewhat personalized, even in patients that are getting bombed with antibiotics and getting vancomycin. So it's not entirely predictable. So I think there are different levels to sort of systematically try to understand the whole idea of having consortia, because it's not just simply what is in your concoction, but it's actually what it's actually doing in the host. And part of our readout, I think, is engraftment, which again, as I'm sort of mentioning, we don't really fully understand the biological basis of that yet.
So Gary, just a quick follow-up on that thought then. How important is the method of administration, enema versus capsule, and matching disease indication to tracking engraftment? So are different diseases, is there any data out there to show that different indications might be more engraftable than others? And again, the administration route question.
I'll give you my own bias on it. And again, it's just my own opinion on it. First of all, I actually think that the route of administration is probably important. If you give it orally, then it's going to go to the small intestine with bile and pancreatic acids and pancreatic enzymes. It's going to be in a completely different environment unless you protect it all the way to the end of colon. The initial environment that your organisms will actually encounter will be different than enema versus delivering it orally. Fortunately, I think for Clostridioides difficile infection, it's actually very sensitive. So you can give it either route, and there's a little bit of a difference in where you give it an enema or you give it orally or colonoscopy, but it still seems to be quite sensitive. I don't know what that would necessarily be in other types of diseases, you know, so I think the route of administration might actually sort of be important in that.
Hervé, you might have some experience with that with your trials.
Yeah, indeed. In fact, it really also depends on the indication. If you want, I mean, you are taking colonic bacteria to produce your drug. They are not ileum bacteria, they are colonic bacteria. So if you reintroduce colonic bacteria into the colon, that's a certain effect on the host. And if you are re-administering bacteria that are from the colon in the ileum, that would be a different effect also on the patient, on the host side. So it's indeed very important to work with the right scientist to make the right choice. Here, for example, in GVHD ourselves, because we want to calm down the activity of the immune system of the patient, we are going to the colon. If you want to exacerbate a little bit the immune system, you have to go into the ileum. So it really depends on the application indeed. And there was also a difference in terms of the severity of the disease, because when you go enema to capsule, you lose some density. And in some density being the number of microorganisms per milliliter, and sometimes you want to have a bolus effect. So you have like no choice but the enema if you want to have that bolus effect. But apart from that, it really depends on the indication. Yeah.
So this leads us right into some of the Q&A box questions. And this is from Lynn McFarland. In capsules of donor stools, how strict are strict anaerobes surviving if they don't produce spores? And also how much of the anaerobes survive through frozen preps? Does anyone have any data on that?
So what I can tell is that there have been studies. We are not, when we are using frozen microorganisms, we are not just we do not just put them in the fridge. We are using cryoprotectants, and there have been quite good data about the efficacy of this type of approach to preserve the bacteria. Of course, you lose some of them, but globally, you are able to save most of it. The other aspect is that when we are dealing with tools, we are not dealing with pure culture. We are dealing with a matrix. And this matrix is indeed protective for the bacteria. And you can see that with, for example, very sensitive bacteria that you can only grow in the anaerobic chamber. You put them out for a few minutes, and then everything is dead. If you take, for example, germ-free mice that you colonize with the same bacteria, you take the stool from these mice, you can keep them in ambient air for days, and then puts back the stool in the anaerobic chamber, and the bacteria are still alive. So the matrix of the stool is active in protecting the bacteria also.
Thank you. And then following through, this is a really fascinating topic I think the audience is picking up on. So the next question is about prebiotics as a complement or supplement to FMT. Do any of the panelists want to comment on, is this going to be a real option to improve treatment efficacy? And should we focus more on the donor or the recipient side of this kind of prebiotic induced efficacy?
I can answer that if you want.
Yes,
basically,
please.
Prebiotics are food for the good microbes we have. So it's in one way, it's coming back to the diet. So of course, if you have an healthy donor who eats a lot of vegetables, he will have a better microbiota with higher diversity, better bacteria in it, and so on. So of course, having this type of donor is probably good. I say probably because we don't know yet. This is really ongoing research. And on the other hand, as I mentioned, and it was mentioned by many others, if we want to stimulate the good microorganism we are giving to the patients, obviously stimulating them with fibers is also positive, and it can be done with diet also.
Yeah, and I can build on that. Want to really echo what Harry said, and I think diet does matter both for donors and recipients. Again, in cohort studies, we've seen if patients have a sufficient or a higher fiber diet over 20 grams a day, they have better responses to immune checkpoint blockade, with significantly enhanced progression-free survival to anti-PD-1. We actually are running dietary intervention trials where we're giving either high fiber or ketogenic or other diets to patients, and that's either through our food kitchen, which is where there's packaged meals, which we actually have the patients come in and pick up. But there's more simplistic dietary interventions. Carrie Daniel McDougall is basically administering 1 cup of navy beans per day to her patients in a cohort of colorectal survivors, and she's run a study where she looks at the microbiome and then also other variables in those colorectal cancer survivors. So I think, yes, prebiotics will definitely play a role, but they're not all created equal, and we don't know which ones are the best and which are patients can actually be compliant on, and then which ones can we put in a pill is the big question too, because can we actually engineer a prebiotic that we could put in a capsule that could enhance, at least in patients with what we consider to have a favorable microbiota, to enhance that microbiota, and then in patients who have an unfavorable microbiota, can we actually manipulate that and then use prebiotics in conjunction with that?
Thank you. And you're right, they're not all created equal, and there may be some surprises. And Jen, I think you had some interesting data a few years back on better responses from obese patients, which maybe, we don't consider those as healthy recipients, but maybe the obese microbiome has some sort of profile that increases efficacy for the immune checkpoint. Can you comment a little on that study?
Yeah, exactly. And so Jen McQuaid has done a lot of work on obesity in response to checkpoint blockade, noted that particularly in males, obesity was associated with better responses to treatment with anti-PD-1. Now, is it the microbiome? Is it hormones? It's probably more likely hormonal influences in estrogen, and that at least in patients with melanoma, we've seen that testosterone and androgen receptor signaling can be deleterious, so I think it's more on that front. We're trying to understand that a bit better, and we actually in a recent larger cohort explored obesity, the microbiome, didn't see, in BMI, didn't see that many differences, and it seemed to be less at play, in our larger cohort of checkpoint treated patients. But nonetheless, I think, like some of the diet data that's coming out now, like some of these diets that in patients are, embracing them, some of them may not actually help and may harm, and certainly we've had data on over-the-counter probiotics and how they might not help and may actually potentially harm responses to immune checkpoint blockade and other variables.
So I think the pro and prebiotic supplementation needs to be, carefully considered. Not all of these formulations are created equal, and we don't really have good information on what's going to work and what doesn't. But it's certainly an area worth considering.
Yeah.
So Phil, I just want to jump in real
please.
quickly. And so we only have maybe like 15 minutes left or so. And so I know there's a question from the audience and we can kind of move on with that, but we've talked a lot about donors and we are thinking about standards for a process that is very complex, right? So we've basically started at step 1, talking about donors and how can we do that, and the administration, talking about enemas or a capsule form. And so something else I would like to think about too is the types of measurements we're making. So Vincent gave a really nice talk about using flow cytometry. Most people have discussed metagenomic data. And so I think just talking a little bit about standards for things like bioinformatic platforms, is that something we can standardize, or is there a standard method? Should everybody be using a specific DNA extraction kit? I think that starts to get into kind of muddy water as you think about advancing and improving technologies. So Phil, I don't know if you want to start with the audience question and then we can kind of go from there.
The first one that I think pretty directly questions or opens that topic. What are the promising measurement approaches to move away from just taxa profiling towards functional effects of microbiota in healthy communities? Is it whole shotgun sequencing, proteomics, metabolomics, or something else?
I'll take a shot at that as an initial comment.
Sure, Gary.
So, we just like everybody else started with shotgun metagenomic sequencing. That's only part of the answer. And I think that a multi-omic analytic approach is probably going to be the most feasible, because I think each one of the platforms has its strengths and it has its weaknesses. I think that when thinking about functionality, looking at taxonomy, but also the metagenome, thinking about the structure of the various types of genes, relative abundance of genes intersected with metabolites. Production is actually something of significant interest to us and other individuals. And that's where diet, I think I sort of favor the notion that the metabolites are actually more sensitive to diet than actually the taxonomy of the microbiota, which is, at least in our hands, and other people have shown that it's actually very individualized and it's not a profound effect. But the metabolome does respond fairly significantly to various types of dietary inputs. So I would say looking at a combination of at least shotgun metagenomics and metabolomics may be giving a better view of structure and function.
And something here. I completely agree that I think multi-omics is the best approach to have rather than focusing on only one approach. However, because we have been discussing how complicated it can be, and that we can't reproduce the results or we cannot apply to different patients, shouldn't we have always a proof of concept, like try to, whatever we find with the multi-omics approach, try to reproduce it in the lab to make sure that it's true, we really found the bottom of it. Does that make sense?
Yeah, I think that goes with the next question, which is, can we kind of customize donor material for indication? So for example, there are efficacy specific considerations to be made if you're trying to improve immune checkpoint therapy versus if you're trying to radio protect or radio sensitize the microbiome to radiation therapy, or such as Vincent's work on defining what the immune activation is after transfer. So I'd like to hear some comments on kind of indication-specific customization of FMT. Is that possible? Is that a goal we should aim for?
Maybe I can comment on that. The microbiota, of course, when we talk about it, it's like that. It seems that it's one entity with one mode of action. But of course, it's not the case. There are plenty of mode of actions, plenty of effect of different effects. And as all the diseases are not related to the same impairment, it makes, I think, perfectly sense that some donor might be more prone to induce therapeutic effect in some disease and maybe not in others. So from my point of view, it's clearly not one stool fits all.
Right. And yeah, I'll jump onto that one too. I think, where's the bar, right? I think for C. diff, the bar, relatively low, right? I think same thing for immune checkpoint associated colitis. Candidly, those patients have a very dysbiotic, low diversity, gut microbiota. And so you can actually give them a healthy donor FMT, and most of those patients will actually have therapeutic benefit from that. The bar is higher, and I think others would agree for IBD and some of the more complex diseases where there's a lot of other variables that are contributing, and cancer is in there, right? And different types of cancer on immune checkpoint blockade. Is the bar going to be at the same level, or is it going to be at different levels? And so, yeah, I don't think it's a one stool fits all either, but I think they're probably ultimately, could we have a universal ideal donor recipe, if you will, someday that would enhance, kind of overall immunity, response to immune checkpoint blockade, reduce toxicity. I mean, I'm a dreamer, so, I think it's out there, candidly, and could we ablate microbiomes and transplant that in, and everyone would be happy and healthy, maybe, but I think it's certainly laudable and it's a good goal to strive for, but I think we have to live in the present and know that, there's going to be different recipes.
But what would be the strategy to select this perfect donor, other than serendipity? Or I understand that diversity is not enough. So how do we get it? Because right now it's just a feeling that it's serendipity, and sometimes we get a good one, and when we jump on it, it's just a partial question.
Well, I think like looking at the different cohorts, right, of diseased versus healthy, then looking at the, different indications of FMT and where it's been effective, and not at taxonomy, but looking at the functional attributes of these favorable microbiota that have favorably impacted response, to whatever disease state and are associated with better overall health, I think would be, but we need to compare it across different disease states, not solely.
I think, Jen, that's exactly right. There are probably some universal considerations more on the safety side of things, taxonomic profiling and SPF and probably 16S sequence data. But then the efficacy side of this would be really indication specific. And as Harry said, not all stools will be applicable to all indications. So I think different communities are going to have to be defined by how they're going to be used. And there's a great question in the box that kind of follows this topic, if I can introduce it. And this is from Lucille Plurdy. Sorry if I mispronounced that name. But it's relevant to donor collection and sampling again, and selection. So has there been any effort made to collect samples from isolated non-industrial populations? There are several projects out there biobanking across the world in some of these isolated populations. And have any of these communities been tested for FMT efficacy?
I guess it relates also to the question of the diet, because I mean, if you use this kind of material to perform FMT to industrialized Western diet, to Western people, the diet will not be adapted, and so you will lose the benefit. You will quickly lose the benefit of using this material to perform FMT. I mean, that will be my opinion, but
I know Justin Sonnenberg from Stanford has done a lot of work in African populations defining the diversity of those communities, the gut communities from those populations. I'm not sure where those samples are banked and if they can be tested for efficacy to modern industrialized disease, but it's an interesting thought.
In the donor selection, I think with the current process we have and based on the criteria we have, they might not be eligible.
But there would be a good source of new commensal species that you can try to isolate and cultivate and then understand the interaction with the host. But you could not use this material for FMT, I think.
Yeah, I mean, I think it's a very intriguing question that could be tested in animal model systems, but in humans, I think there are enormous hurdles to be doing something like that.
One more question from Brian Roth. Is there a concern of monoculturing the microbiome if we standardize in that way. So again, this is a diversity question.
I mean, one of the questions of growing the microbiome is how stable it will be with time, I think. But I know, for example, that in Norway, they are using this kind of treatment in a traditional way. They isolated stools from an healthy donor, I think it was in the 80s, and they grow it, and they use this material to treat C. diff infection, actually. And you can even buy it on the web.
So I think Sarah will want me to ask this last question that we had formulated. So, but it's a very direct question. If we were to set up a working group to develop standards for FMT, what should the top priority be? Should it be something like donor selection or patient exclusion criteria, engraftment monitoring, GMP to get the material, or something else? What should be prioritized for developing standards for FMT?
And we have to pick one, Phil?
No, you can give them in order if you want to.
Yeah, exactly. Exactly. And it was shown before, it's like, we put out this position piece on considering gut microbiome modulation in patients with immune checkpoint blockade that we published in Lancet Oncology. And there's just so many variables to think about. But I think like it does highlight, before, during, after, all the considerations, both donor and recipient. There's so many considerations. But I think, like we brought up, what are the standards for sequencing, not only 16S, but moving beyond that for metagenomics, for metabolomics, for the profiling studies, but then the intervention. I think there's a lot really to tackle there. But I would love to hear others' thoughts, Harry and
Jennifer,
others.
could you? I'm going to put you on the spot. Could you give us that citation again, or maybe put
Totally.
it in the chat box?
Yeah. Absolutely.
So
It was
much.
shown in a slide, and I took a picture because I was proud, like a proud mom. I'll pull it up.
Thank you.
You bet.
Any other comments on priorities for standardization of FMT?
I think today we are working a lot in silo, and I think the first element is that we can work together, because I'm aware myself, I'm part of what we call the Pharma Biotic Research Institute, and we are only few working on standardization within that group, and there's other groups. So I think the first element would be to work together. And if I were to pick one from your list, personally, I think we should be defining how we monitor engraftment and everything, because not only it will help for academic research, but also for the regulatory, because I think there is a key element. I'm not saying that when you engraft, you respond, but we know that if you don't engraft, probably you do not respond to the treatment. So I think a key element is to work on that kind of definition and how we monitor it, and that will also inform the regulators, which I think is very important.
Thank you. I will put Chrissy on the spot here. Chrissy, are the Bray similarity measurements enough to do that engraftment? To monitor engraftment that way? If we get the recipient similar enough to the donor, is that going to be a good standard for us? And then what cutoffs would be used? Does it have to be 90% engrafted or similar?
So I think there are two different topics here. One is for using the standards that we're developing to make the pipelines we use as better as possible, and so we can have confidence in them. And I think once we gain some more confidence on what exactly is in our FMT or what exactly is in our recipient, maybe we can get closer in regulating better this process. And yeah, as I said, what we aim is to create a range of how close they have to be, like how good your pipelines have to be, because setting a number or giving a specific, well, do this DNA extraction or follow this pipeline, I think it limits a little bit innovation. What we want is to encourage innovation, get like, hopefully pipelines will get better. So that's the one part. I hope I covered it. Second, like regarding getting closer the recipient to the, sorry, the donor to the recipient, I don't know if it's gonna solve the problem of seeing the differences that some.
Or is it a good way to measure how engraftment is going? I mean, this can be done by 16S profiling as well, but I just was wondering if the similarity indexes were a good way to also monitor engraftment.
The problem is that actually we don't know. As Jennifer mentioned, for C. diff infection, it works whatever the microbiome you use. And I'm pretty sure if you use the microbiome of your dog, it will work also. So it really depends on the indication. It is also likely that in some indications, such as C. diff, maybe colonization engraftment is not necessary. Maybe it's related to bile acids that you have in your preparation. So I think it's really, the issue is that it's really beyond the FMT field. It's really about microbiome field in general. When you are doing metabolomics on your stool, you are able to identify 10% of the molecules. So how are you going to standardize that? I think probably we have to acknowledge that FMT is not a drug like the other one. It probably needs some kind of specific regulation, specific status, because we're currently and probably for many years, we will not be able to define precisely what are in this tool.
Harry, your comment leads right to the last question in the Q&A box, and I'll direct
You have like
first.
1 minute for answering.
Yes,
We're already over, so 1 minute.
Oh, I had 5 minutes left. I'm sorry.
That's a sign of a good
case,
webinar, good conference.
That's true, and a really robust discussion, and I appreciate that. So let me get this last question out, having to do with donor samples from, and Gary, you probably are in a good position to answer this, from family members, because as far as matched genetics and other factors are concerned, and also ruling out donors with any disease, do you have any information about using family members for donor material, and is that better than pooled or banked?
You know, I think that the notion that families could be in the same environment, maybe that would make a difference. I'm not aware of really strong information saying that if you're a family member, it's going to be better than anybody else. I think our current standards are probably a reasonable thing. I just wanted to comment about the engraftment issue. I think it's a really important issue. And I think it's not just 16S. I mean, I think you have to get down to the strain level and actually figure out what that organism is. And are you engrafting in the stool, or are you grafting at the mucosal surface? I think that really trying to understand that basis, as Herb had sort of mentioned, if that is tied with biological responses, I think that that's an important thing to be looking into.
All right, so thank you everybody, for all the panelists and the speakers, as well as the audience for all of their questions. I think this was a great discussion today. You know, we heard a lot of challenges. I don't think this is going to be a simple solution to move towards standardization, but we also heard a lot of successes. So, you know, therapy, FMT therapy beyond C. diff is really a direction we're heading. And I think a key takeaway from this that has been brought up by several of the panelists is that really collaboration is going to be key to addressing a lot of these. So having meetings like this and follow-up discussions to really start to attack some of these priorities kind of one at a time and see where we can go. So with that, I'll just ask everybody to provide feedback. There will be a brief follow-up survey, and there's going to be a link in the chat box, but we'll also send that link in an email. So if you have thoughts about where we should start, you know, for developing FMTs or a working group, please put those into the survey.
Thank
Okay.
you all.
So
Thanks all.
I think I have now the pleasure to, or the unfortunate pleasure to close this very impressive meeting. I am not at all from that sector. I'm a virologist by training. So, but it's really passionate, and I think you could continue still for a couple of hours, bringing in more and more information. And I hope this is only the first that we organize, because I have the impression a lot has still to be discovered, but I congratulate all of you with apparently a huge progress in this promising field. I like to thank two persons here. I like to thank Philippe Sabot, the Executive Director of IABS, who continuously supported and proactively kept this project alive, organize it. And last but not least, a very, very warm thank to Sean Novick for creating, initiating, and persisting with this project. I think I hope the most difficult has been done with this first webinar, and that many other meetings will follow. I'm very thankful to Sean for having opened this discussion and this opportunity for IABS, because it's really within our remit in order to bring all of you together, and probably others as well, in order to not only find the actual sciences, make the diagnosis of it, but particularly to see the challenges in order to bring the therapeutics and the prevention tools that can come out of it to the most, the largest possible amount of people. So I look forward for your successes, and here I will close definitively this passionate meeting, which again could go on for hours and hours, bringing interesting information. Thank you all for participating, and a lot of people staying on board till the end. I wish all of you an excellent weekend and hope to meet you soon. And I like to thank Abby and Maddie for the logistics behind all this. Bye-bye, keep care, and hope to see you soon.